in the last session for the second day. Today we have a very interesting perspective on organofluorine compounds. It is about pharmacoinformatics studies on fluorination chemistry. So for this, we have a very appropriate expert who has a vast knowledge in this area. And you all know Professor Bhartam personally, but I still would like to, because I have few students, we are from Punjab University, so I really want students to know about Professor Bhartam and his immense contribution in the field of pharmacoinformatics as well as medicinal chemistry as well. So Professor Bhartam is a very good support for me as whenever I think of NIPER, before even when I was at Punjab University, I never faced any problem because I also did one GAN course here in 2017, and whenever I approach sir for anything, he's always available. Not as I'm currently at NIPER, but even I was not at NIPER, and he's always ready to help for any academic activities. He's always available. So so really thank you, sir. So sir completed, got his PhD. Actually, he did masters from Vishwa Bharati, Shantiniketan, and then did his PhD in 1990 at University of Hyderabad. So his research experience lies in the organic synthesis and computer-aided drug discovery, which he is doing at NIPER over maybe around 24 years, 2001 onwards. Then molecular modeling of metal-based drugs. He did this work at Philips University, Marburg, Germany, for 1 year, 2002-2003. Theoretical organic chemistry. So he worked at Guru Nanak Dev University for about 7.5 years before moving to NIPER Mohali. He was a postdoc at University of Alabama, USA, from 1991 to 1993. Applied theoretical chemistry. He was a research fellow at the Hyderabad University, where he did his PhD. He has over 29 years of experience, including teaching experience of 23 years at Punjab University, at NIPER Mohali, and at GNDU Amritsar. So he had, while being a good teacher and a good researcher, he also took a lot of administrative duties. He was the head of the department of medicinal chemistry. He was a head and in charge of Department of Pharmacoinformatics. I think he's the one who started this department itself. He was the associate dean academics. He was a dean at NIPER, our institute. He also was officiating director for a short span of 6 months. Then he got several awards and honors. He has a fellowship of Indian Academy of Sciences. He got OPPI Scientist Award in Medicinal Chemistry. He also got Ranbaxy Research Award in Pharmaceutical Sciences. Chemical Research Society of India Medal he got in 2008. He got Fellowship of the Royal Society of Chemistry London. He is a IBM faculty. He got IBM Faculty Award, and he's a recipient of the Fellowship of Alexander von Humboldt, on which he worked at Bonn, Germany. So he's a member of Research Council of CSIR CDRI Lucknow. He's a member of a Research Advisory Committee of ICER Mohali. He's a member Editorial Advisory Board of Current Computer-Aided Drug Design, member Editorial Advisory Board Indian Journal of Chemistry, and member of many funding agencies like DST, CSIR, DBT, and their research council. He has supervised 49 students. Then he has MS Pharm students 225, and I think this number is not updated, so it is he has guided more than maybe 225 students. A few master students also maybe at GNDU 16. Several research fellows worked in his lab 12. Then several research visitors continuously visited. He got several grants. Number is big. Then he has total 385 publications to his credit, which has over 315 international research publications with an H index of 49. He has published almost all the good medicinal chemistry publication. I just wanted to list few, and because master students, I'm doing it only especially for first year master students, because I wanted you to have just such kind of dream to build this kind of CV. So you see, he has published in Bioorganic Medicinal Chemistry many articles, European Journal of Medicinal Chemistry, Bioorganic Chemistry, Drug Discovery Today. Then he has Chemical Review, Chemical, and so the number is more. I'm just listing the journal name. Then Journal of Organic Chemistry, ChemCom, Organic Letter, ACS MedChem Letters, MedChemCom, ChemMedChem, JMedChem, and then there is a JAX, an Angovanti Chemi, again many JAX. So I think really, sir, I think you are an inspiration to our department and to all the students and even to the faculty. So with this, I would like to request Professor Bharatam to deliver his talk. Yeah. Professor Arpan is here at right time. We just finished the introduction.
Well, Professor Salunke has got a lot of time to read my CV. Okay. Well, anyway, today my topic is on the pharmacoinformatics studies. I'm really happy that this conference, this workshop, we are discussing the entire science with reference to not only pure organic chemistry, synthetic perspective, but also with reference to physical organic chemistry, energies, conformations, and many other structural details. This is all part and parcel of our work in our lab. So that we have learned many things to learn, and I hope my students are enjoying all that. Now I would like to start with this comment which I like the most, and I tell all my students so many times. This is title of this article published in JMedChem is What Makes a Great Medicinal Chemist. So this Professor Murko, who is from Pfizer, he listed about 18 or 20 different characteristic features. Out of that, a few of them are most important. Out of those most important things is about thinking in 3D. That's what I also do that, and whatever achievements we had in our lab are mainly because we cultivated this habit of thinking in 3 dimensions about the chemical species. So I wish you to read this article, and this is worth reading for any medicinal chemist, whether he thinks in 3D or not. Okay, I took this idea because drugs are 3D objects, macromolecules are 3D objects. The interaction between drug and macromolecules happen in 3D. The dynamical processes are 3D, chemical reactions are happening in 3D. To help in understanding all that, we have created a department called pharmacoinformatics, where we collected lots of software and then we teach everybody how to think in 3 dimensions. The information technology we utilize extensively in this department for the drug discovery, drug development, drug toxicity, and other studies. We also use artificial intelligence technologies in this department. Having said that, I would like to add that point. When I started my PhD, none of these facilities were available. As a result, we needed to do everything in our mind. Now these facilities are available. You can use on your mobile also. 3D structural visualization and analysis is possible. So do adopt this habit. It will help you all. In pharmacoinformatics department, we do study all these varieties of concepts we adopt. QSAR is one which is extensively being used as a bridge between pharmacology and the chemistry. Then molecular docking, dynamics, virtual screening. In addition to all that, we also use quantum chemistry to understand various aspects, de novo design, chemoinformatics, bioinformatics, and all this, including artificial intelligence. 3D thinking in fluorine compounds. There are so many of these compounds are known already. We discussed many of them, but we are sure that if we know how our fluorine atom is embedded inside that architecture of the chemical compound in 3 dimensions, we do get much more clear idea. For example, look at these compounds. 2D structure as well as 3D structure has been provided. If the fluorine is at one end, we are okay. This is a green color is fluorine, but light blue are fluorine. We can probably easily top, okay. Here, these are the 3 fluorine atoms in this molecule. But that's probably very straightforward. We don't have to think too much. But if we have it in little more complicated manner, we do need to think CF3 is okay. CF3 here also probably is okay in this molecule. If we go deep, we need more clarity. For example, in the next slide, I'm showing this is a 1D information about this compound. And then here this fluorine is in 2D it is very clear, but look at the 3D, where is it? How it is embedded. When it is in 3D, we can rotate, we can zoom, we can represent it in many ways. CPK models, for example, are also very useful. Likewise, there are new perspective is available when we think in 3 dimensions. So that's what I encourage all of you to adopt. Here, this is the light blue. No, this is green one, yeah. Right. So think in 3 dimensions will help you all. Anyway, let's go to the next slide here. So this topic of pharmacoinformatics we are applying in many fields. We started this as a topic of integrating bio and chemoinformatics, but we are applying daily for the drug discovery, drug delivery, drug metabolism, drug toxicity, and every field of pharmaceutical sciences we can apply information technology. Artificial intelligence can also be applied in every field, including drug delivery, drug toxicity, everything and anything under pharmaceutical background, maybe even pharmaceutical management, people also should be able to utilize this. So there are other names for the same topic. There are several synonyms. I don't want to elaborate on this. But I would like to tell that it is not difficult, especially for us, because our teachers in high school are pretty good. They teach very good mathematics. Compared to, sorry to say this, compared to American universities, American high schools, Indian schools teachers teach pretty good mathematics. Much more intense mathematics in high schools in India. So, so that is a good advantage for all of us. And so let us make use of this and get benefit. Informatics in pharmaceutical sciences can be 2 types. One is the science-oriented, the other is service-oriented. That means I want to say that pharmacy informatics, clinical informatics, healthcare informatics, there are so many varieties of databases, so much analysis is being done, and so much is practical. So how do we use all this? This is a flow chart to show how the data flow happens in drug. Once the drug is identified, before that, so many information technologies are practical. After the drug is identified, so much more is possible in this. So bioinformatics, some of you know about it. It's over 30, 35 years. It has become very popular, and mainly we use it for target identification and target validation. Whereas chemoinformatics we use for analyzing the data, chemical data in many ways. Virtual screening is one of the important possibilities, and for this purpose of this symposium we have Fluorobase is a database available of all fluorinated compounds. It is free of cost. You can download and use it at any time about these species. Then ADME informatics is also practical. Yesterday we discussed about this, and there are so many. We can optimize the ADME properties. I will give one example a little later. For toxicoinformatics, there are so many varieties of tools available, like TopCat, Derek, Azure Expert. We can use any of them. For example, let us go to the day-to-day life where we are using fluorine. Every day, do you have sodium in your toothpaste? A big advertisement you have seen. I think when you were in class 5, 6, this was very popular in TVs. Now I am asking the question, do you have fluoride in your toothpaste? Inorganic fluoride. So there are so many compounds, sodium fluoride, SnF2, and all these species are generally available in our toothpaste. And these are all general inorganic variety. But how we are using actually? We are using to clean the bacteria. That means the bacteria which is settled early morning when we get up on our teeth, we want to remove them with the help of fluorine. This is it not biochemistry? Is it not enzyme technology? Are we not killing the bacteria? Yes, we are. So for us, it is a biochemistry topic. If a toothpaste is having fluoride ion. Similarly, let us look at the organic compounds. So many varieties of organic compounds. We listed many of them yesterday and today. Many of them are already approved drugs. Implications are many varieties. Some of them are anti-cancer compounds. Some of them are anti-fungal, antiviral, and antibacterial. Many varieties are already known. So this I don't want to elaborate. But now I want to go to a little more close. CF3. CF3 is present in many drug molecules, approved drug molecules. Again, anti-cancer, antibacterial. We can list many of them. Now, a few more are O-methoxy. Not only the CF3. For example, this was partly discussed earlier. Bioisoester for methyl. Many of the organic chemistry people are using. One more advantage with this is that if we have a methyl group and you replace with CF3, you will get a big IPR. Intellectual property is practically possible. For commercial perspective, this is also important. So many people are using this. Enhanced metabolic and chemical stability is practical with methoxy trifluoro compounds. Increased steric protection, high lipophilicity, improves permeability, reduced hydrogen bonding interactions, improved pharmacokinetics and bioavailability. These are all listed for the compounds with OCF3 group. First I showed fluorine, then I showed CF3, then I am showing OCF3 compounds. All these are available in these publications which we referred yesterday also. So, but then what is pharmacoinformatics doing with this? That I will come to this. Yesterday, Professor Altman has already described what is hyperconjugation and negative hyperconjugation. Let us consider this one more time because this is somewhat deeply involved topic. Hyperconjugation is a topic when a pair of sigma electrons are participating in delocalization. It is hyperconjugation because sigma electrons are donated. Negative hyperconjugation is when sigma bond is accepting electron density from the nearby. This is negative of the original hyperconjugation. That's why it is called negative hyperconjugation. This definition has been given by molecular orbital people. Other than that, this is already known in the glucose chemistry or sugar chemistry. It has an anomeric effect. Yesterday again this example came up, and so anomeric effect which we learned in standard organic chemistry books and the negative hyperconjugation we learned in advanced research articles. This slide, actually, this equation we have seen yesterday. You see what happened. Here the orbital overlap becomes very practical here. The lobes, the two lobes here overlap very much. What is here? It is a sigma star orbital. Between the carbon and the fluorine, it is an antibonding orbital. When electron density from this center is pumped into this particular antibonding orbital, the carbon fluorine bond becomes weak and then fluoride ion gets out. This was what I was explained yesterday. The same thing, I am using the same equation here. But we have to be somewhat careful. If a fluoride ion is floating in the body, it is probably not very comfortable situation. So, for example, this compound is only approved for topical use. Probably because of the fluoride ion in vivo may not be very safe. Now let us go to this question. What are first order interactions in chemistry? Sigma bond is first order. Pi bond is also first order. Then there are second order interactions. Just we already discussed hyperconjugation and negative 2 hyperconjugation are second order interactions. How do we understand them? How do we evaluate them? There are push-pull influence. Under some conditions, fluoride ion pulls the electron density. Some conditions, fluoride ion can pump electron density into the given system. How do we analyze all that? Can we get the result quantitatively? For that, there is quantum chemistry. Quantum chemistry helped in the form of NBO analysis. It provides a shortcut to analyze this. We can estimate the energy due to a particular second order interaction very easily. We can analyze the first order also, but that is much easier to do. Second order is a little more tough to analyze. For that, this NBO analysis helps. What we need to do is that we need to obtain the wave function. To obtain the wave function of a given drug molecule or any organic molecule, we need to solve the Schrodinger wave equation. When we solve the Schrodinger equation using either molecular orbital theory or the density functional theory or any other advanced theories like coupled clusters or configuration interaction or many body perturbation, when we use all that kind of quantum chemistry, we will come across with something most standard that is called wave function. We identify a very well defined wave function for a given compound. That wave function carries lots of information. Almost every information that we want to extract is a part of the wave function. That is postulate 1 of quantum chemistry. Using that, we need to supply that wave function from quantum Gaussian calculation or any other software to this particular NBO software, which will analyze the various interactions. And the second order interactions can be estimated like this. Energy due to a given specific second order interaction can be estimated like this. It is due to charge, that is the total orbital occupancy into the Fock matrix, square of the Fock matrix element, and energy difference between the electron donating orbital versus the electron accepting orbital. This is a very simplified equation which is helpful in interpreting the wave function to understand what is the extent of the second order delocalization that is happening. How much negative hyperconjugation is there? What is the energy associated with it? Is it 0.2 kilocalories or 25 kilocalories? There is how much importance we need to give to in a particular environment for the second order interaction. To analyze all of that, there are these facilities available, and students in our institute, they are utilizing this information almost every day. The role of fluorine in pharmacokinetics. For example, here, look at this example. The modulation of pharmacokinetics is possible by introducing extra substituents which carry fluorine. That was partly discussed with a couple of examples earlier. I am showing another example here, which was part of our. For example, here there are several pharmacophoric features. Here, anti-cancer based pharmacophore, for which one feature here is hydrogen bonding feature. Another feature here is due to the fluorine center. Hydrophobic moiety is here. So there are so many of these which can be further modulated with the help of additional fluoro group introduction. This is helping in enhancing the metabolic stability as well as the membrane permeation. This was published recently in 2021. Now let us take this example which we have pursued recently. There is a target called FTSZ. This is an antibacterial target. It is a protein. The protein gets polymerized. The polymerization of the protein, if we can stop, then the bacterial growth can be stopped. Because once the protein gets polymerized, that means the FTSZ protein, if it is polymerized, it forms a linear chain, and from that it forms filaments, and these filaments actually are bound to the cell wall and they cause cell division. If the cell division is happening fast, then the bacterial cells grow at a faster rate. If I want to stop that, I can inhibit FTSZ polymerization process. For example, Staphylococcus aureus FTSZ polymerization. For that purpose, this particular compound has been identified as a prodrug. Is a difluoro species here. Now, this compound is known as a prodrug, and it can be bioactivated to give you this particular drug. This compound is known as PC190723, a very active inhibitor of the polymerization. However, they found that it cannot be made into a drug. When it cannot be made into a drug, what modifications are required? It is mainly because of the metabolic instability originating in this particular benzethiazole or pyridothiazole fused species. Then, due to this corner, metabolic stability is very much less. As a result, can we modify this? The idea was that can we introduce CF3 group in place of the CL. That is very much practical. Lots of examples were already available. As a result, it found that this compound, this chloro species, may be getting converted into the OH group due to this reaction, dechlorination followed by monooxygenation. The same thing does not happen when you have a CF3 group here. So TXA709 is a better prodrug because it does not undergo this reaction. That means metabolic instability is removed, but metabolic stability is introduced, and this active drug is TXA707, which is obtained by simple hydrolysis. Look at this. This is a species which can easily undergo hydrolysis, and it will give you the ortho and para difluorobenjamide. This is a pharmacophoric feature. This pharmacophoric feature is required for the drug action. What is the drug action? It is the prevention of the FTSZ polymerization. So this kind of simple modification helped in realizing a new prodrug and the new drug. It is still in clinical trials. It has not been approved yet. And so what to do with this? We can do molecular docking, understand the 3D structure of the FTSZ protein and how it is showing the drug action. This is a protein. There are two sites of action. One is the GTP binding domain. GTP binding domain is has been suggested as an important site of drug action. Another site of drug action is the IDC domain. It is known as, what is IDC? Come on, who is working on IDC? Interdomain cleft. Okay, I don't remember all the time. So interdomain cleft. It was suggested that this compound may be binding to either GTP domain or the IDC domain. So in that context, if we do molecular docking, this is a powerful pharmacoinformatics technology. What we did, we docked this compound and all other related lead compounds inside the GTP binding domain as well as in the IDC domain. It has become clear to us that when the compound binds to GTP binding domain, inhibition is not sufficiently high. Inhibition is pretty good, and in future lead compounds we should design by exploring the binding pose of TXA707 inside the IDC domain of FTSZ. That is exactly being done in our lab, and based on that, our students have found a few compounds which they will not tell me that is secret. I will not tell you right now, but they have found compounds which are nanomolar active compounds for antibacterial effect. That secret will come out after patent publication soon. But only thing is that their fluorine is not there in our lead compounds. We will explore that in the near future. Okay, now let us look at other examples where naturally occurring organofluorides. In nature, are there any organic compound? Are the plants producing fluorine containing natural organic molecules? When we want to understand that, generally the perception is that nature does not incorporate fluorine in many organic molecules. But there are a few specific examples like this which are available, and then fluoroacetate is a major component. This fluoroacetate happens to be a dangerous compound. It's not a very favorable compound for consumption. So humans, they know which plant sources are poisonous and which are not. But animals, they don't know. They do consume all sorts of leaves. So if at all animals consume these kind of plant sources which contain these chemicals, they finally produce this particular metabolite, and this metabolite is leading to their death. So some plant sources are causing death to the animals because of this most dangerous. Inside the plant metabolism, this is okay, this is safe, because this particular species produces acetic acid inside the plant. There is sufficient mechanism is available to get rid of the CF bond inside the plant, but in animals it is not available. As a result, this species is poisonous to animals but not to the plants. So plants are happily surviving. So look at which compounds are there. Here fluorocitrine. This active reagent can interact with citric acid and form this kind of a complex. This fluorooleic acid is one, and fluoroacetaldehyde. Of course, these two are very much related to each other. And then this kind of metabolites are also found, and many adducts like this were also noticed, and several, not too many, a few specific. These were all reported in this particular article. So how does it work? For all that, there is an enzyme available. This enzyme is known as fluorinase enzyme. This fluorinase enzyme, what it is doing is, it is taking this kind of natural products, and then it is cleaving the CS bond and introducing the CF bond. This is the purpose of this particular enzyme. How it is getting the fluoride ion? It is probably getting from the inorganic sources. Fluoride ion is coming from the inorganic source to this particular enzyme, and this enzyme is transferring to the organic substrates. So this kind of chemistry is happening, and finally this not so safe metabolite is being generated, and so we need to be aware of this chemistry whenever we do drug discovery with natural product compounds. And there are some talking studies were done how this fluorine is binding to the enzyme, how it is actually transferring the fluoride ion, and several studies have been carried out. I am not going to elaborate on that. Now the entire chemistry associated with this symbiosis has been described very well. I hope some of us will get benefit, and these are all the ways in which the animals are getting affected. Similarly, some of the chemistry has been described, especially the citric acid cycle is affected when this kind of fluorocitrate is generated under biochemical conditions. So, if there is no fluorine, citric acid cycle is normal, it is happening. If there is a fluorinated agent is present, the citric acid cycle is not taking place, and that is causing the dangerous side effects. So now I will go to the next set of examples based on artificial intelligence. Artificial intelligence is a very important topic. All of you know it is becoming very popular nowadays, and almost every drug discovery scientist has to learn this. This is realized by Naipar Mohali, and we are teaching this for the last many years. Especially now this has become compulsory course for every Naipar student in the coming few years. So what are all the various topics? Machine learning techniques, deep learning techniques, generative artificial intelligence. Likewise, varieties of artificial intelligence technologies have become available. How drug discovery people are using. Now, before going into that, I will explain there are several internal mechanisms. One can write programs, one can write tools, web-based tools are available. We can use many of them free of cost. Some of them can be obtained and we can utilize them, and we can develop our own tools. This is practical and many varieties of things. For example, knowledge-based systems. This is one type of artificial intelligence tools. For example, Google is a knowledge-based system we are using every day. And the grammar checks in our word processing is all knowledge-based systems. So that we are using. Fuzzy logic, it is based on the fuzziness associated with the logic. It is coming from the physics called chaos. Physics or chaos. There is lots of physics associated with that. From that, a few things are learned and are applied to the mathematical logic, and tools have been developed. And then genetic algorithms, from the process of genetics that are happening in our day-to-day, from one generation to another generation, lots of principles were identified, and those principles were applied to artificial intelligence, and several tools were developed. There are genetic algorithms. Then explainable artificial intelligence is one of the latest technologies, because earlier versions of artificial intelligence was called a black box. It does not explain how it arrived at a result. Now latest in the last 3-4 years is to consider only that kind of artificial intelligence which will explain how it has come to a particular result. So this is also being adopted by the drug discovery people. Knowledge-based systems, for example, there should be an inference engine, there should be knowledge base, there should be a fact base, rule base, and lots of things are there. For the last of 30-40 years, this has been in the vogue. Genetic algorithms, we are using them in molecular docking. For example, to find out which pose is to be considered as the best pose and which pose may be neglected when a molecule is getting docked into the macromolecule. To decide, we do use genetic algorithms. So several software which we are using daily, Gold and Glide kind of software, are adopting the genetic algorithm based technologies in the background. So this is coming from artificial intelligence. Next technology is based on the explainable artificial intelligence. I told that there are tools like XAI, referring to the explainable artificial intelligence is coming into picture. This is very latest. For 5 years ago, it was introduced, and then it has got several internal mechanisms like transparency, justification, informativeness, and uncertainty estimation. All those things are coming from statistics, but applied to heuristic knowledge. Thus, it is becoming explainable. Let us take into account how we can use, how we are using in hypermodeling. Let us take a few examples. For antibacterial activity, we are employing them. This fluorinated derivatives and flavonoids and chalcones, all of them are supplied as an input. When we generate an artificial intelligence model, we need lots of inputs. The more data we supply to train the model, the better it is in performing in giving the good results. So we supplied lots of information like this, and their associated antibacterial activity information. When we do that, we can develop several tools for antibacterial research. These many publications have been already reported in the literature. Taking clues, we have adopted for antifungal. Some people have used artificial intelligence, using generative artificial intelligence. And for antiviral also, several groups, people used fluorinated compounds. Now what did we do? Okay, before going there, I have to do what did we do using quantum chemistry. Several of the metabolites are electrophilic in nature. If a metabolite is highly electrophilic, it can be dangerous, it can be toxic. The electrophilicity index can be calculated. A standard organic molecule which has got a omega value, that is called electrophilicity index, about 3 is reasonably okay. If the electrophilicity index is less than 3, the metabolite is safe for human. If a drug is producing a metabolite with electrophilicity index less than 3, that compound is safe. If the electrophilicity of the metabolite is more than 3, we should not use that kind of drug in our drug discovery process. It's a simple, we can use rule of thumb. So what do we need to do? We have to consider the electrophilicity of the drug as well as all possible its metabolites. So for that, one of the major ways of measuring the electrophilicity of a metabolite is to calculate the fluoride ion affinity. If a metabolite is there and if its ability to interact with fluoride ion is of the order of 60 kilocalories per mole, that means the electrophilicity is very small. It's not dangerous. If the fluoride ion affinity is more than 100 or more than 120 kilocalories, it is dangerous. So to measure the electrophilicity, we do this kind of work. We take for example, we take a triazole-based nitrinium ion and we bring a fluoride ion close to it. When a complex is formed, how much energy is released? This is the 2D structure and this is the 3D structure. Here you can see there is strong pyramidalization at this nitrogen center. We calculate the energy released. It is less than 90 kilocalories per mole. That it is less than 100 kilocalories per mole. That means the triazolium ion is not dangerous. But if this triazolium ion is fused inside a cage like environment or perifused environment, again it is increasing, the electrophilicity is increasing. And then if it is fused in this way, electrophilicity is maintained. So we need to know under what condition which metabolite is giving what type of electrophilic metabolite. For that we are using this in everyday research, and these are some of our publications in this year on this topic. Now there is this multimodal deep learning model was developed by a set of scientists, Zhang et al. This is a publication in 2025 given here. So what they did, they developed a deep learning artificial intelligence model called AFCPI. The title of this is fluoro substitution. Fluoro substitution compound protein interaction model. So this is what they called it as AFCPI. Using that, they could understand the activity cliff phenomenon in a set of compounds. Using that, activity cliff refers to a situation in which two structurally similar molecules exhibit significantly different activity values. This is how they used this kind of a concept and developed a model with the help of more than 100 million compounds, 100,000 compounds, and they trained the model across 2,500 protein targets with the help of these kind of the measurable quantities associated with biochemistry. And this model was used in an artificial intelligence network called encoder-decoder. Encoder and decoder. Encoder is to learn from the input and decoder is to provide the output. How does it happen? This is the chemical structure, one-dimensional information of a chemical compound containing fluorine. You can see the very first element is fluorine. And then this is the 2D structure. This is called a fingerprint of the same molecule. That kind of conversion is made before doing the artificial intelligence, and then data is supplied to the model that is the encoder-decoder model, and then when you decode the data coming out of this trained model, you can generate a few new molecules. This was the algorithm that was adopted, and they found that these kind of molecules can be generated. For example, if you have a compound with CH3, if you replace it with CH2F species, then there is a possibility of increasing the performance in terms of the IC50 value micromolar level to nanomolar level. One can convert. This was a suggestion coming from artificial intelligence, and this compound was synthesized, and it was indeed realized that this artificial intelligence model has helped in really designing a set of improved drug-like molecules. Similarly, in this case, CH isopropyl group has been replaced with CME twice EF unit, and then marginal improvement was noticed in this case, and likewise a few examples were given, and they were all discussed in this article. So this is what we did. We took several compounds from Campbell, like 55,308 compounds we have considered. They are all having antibacterial activity from the database. We have pre-screened them for IC50 value, for their drug action details and others, and we choose about these many compounds for initial training. Then after training, we got a model called antibacterial LSTM model, and then we have to, this was a generic model for antibacterial drug discovery. But now we have to fine tune them. Fine tune for a specific protein target, that is FTSZ protein. For fine tuning, we have chosen 572 compounds which were specifically referred as FTSZ inhibitors. From this, we came up with a new model, and this was divided into two varieties. One is the GTP binding type, the other is the IDC binding type. Once we did that, new molecules were generated using CADD. These new molecules were validated with the help of traditional computer-aided drug discovery approaches. For example, pre-processing was done, tokenization was done, then the SMILE notation was created. This kind of one hot matrix kind of matrices were generated from the input, and such data was applied to the LSTM based deep learning approach. Then once we do that, this is the architecture of the LSTM model. This is how the data flow happens in this, and this kind of data flow actually helps in processing. There are several varieties of neurons here. There are several layers are available. Some layers are known as forget layers, and some are processing layers. Some of them are carry the memory from one cycle to the another cycle. So there is lots of detail. There are several details associated with this kind of architecture. Once we did that, we designed these compounds. Our generative artificial intelligence model was employed to design new compounds. It has suggested this kind of fluoroderivatives as the this kind of fluoroderivatives as a result. These compounds were suggested for the GTP binding domain, and these compounds were suggested for the IDC domain. In IDC domain compounds also, we have fluoroderivatives, and for example, the amide group is replaced with this acetyl group. Likewise, a few compounds were designed by the artificial intelligence model. Then we had to validate them with the help of molecular docking, then pharmacophore mapping, then comparing with the physicochemical properties, Lipinski rule and all others with the known lead compound, and finally we could see which are the important interactions which are helping in the improved activity of this class of compounds. Next step for us is to synthesize this and then test them indeed. In another work, this is about anti-cancer drug discovery. The pharmacophoric features can be identified with the help of artificial intelligence, which is hydrophobic pocket, which is the hinge region binding center of the molecule, which is a hydrophobic center too, and which is solvent accessible region. Likewise, we can understand, for example, the green portion is the hydrophobic pocket number 1. All the molecules enter into that. Look at this, fluorophenyl group is entering into the hydrophobic pocket of the EGFR enzyme. And that kind of information helps in a big way in designing new compounds. These results were not our work. This is published work from literature. Similarly, using that, they have designed, they have understood the pharmacokinetics, halogen versus dipolar interactions. They have understood hydrophobicity, electronic tuning, and varieties of physicochemical modifications have been carried out, and the results were published in this journal. Another of our work is associated with anti-cancer drug discovery. Here again we have used LSTM type of modeling. Here this is still not ready for discussion. I'm just showing that this is one result we got, and again we got these kind of compounds suggested by our artificial intelligence model. It says that consider the aromatic ring systems containing these 3 fluorines in this corner of this amine derivatives with CF3 you consider. This is the suggestion coming from the artificial intelligence work that we have carried out. So now I will go to the next details of this fluoro base that is already available. I think I am running out of time, but I will quickly finish. This is a database developed by Mulka and other scientists. This database currently contains over 20 distinct groups of reagents, including methoxylating and fluoroalkylating compounds, with the largest groups comprising more than 50 entries. Likewise, this is the opening page of this database, and this is some of the details. Fluorinated agents database. Data were collected from diverse sources, including literature, reports, crystallographic databases, chemical repositories, pubchem, and other things. And then structure-based research is possible in this database. Smiles notations can be utilized to search, and then additional functionalities for filtering the search is also possible. 3D structures are available, and homolytic and heterolytic bond dissociation energies have been included in this database, and such databases are very much useful for any scientist who wants to carry out research. This database also incorporates the F19 related spectral information. For example, once we search, we can quickly come to these kind of results. Depending on our search query, we will come up with this kind of result. Whether that compound of our interest is present in our list or not, we can easily do. This is a chemoinformatics based. We can use Wiener index, or we can use Tanimoto coefficient. Likewise, there are so many chemoinformatics tools we can utilize in this database. Similarly, fluorobase has been utilized to identify some important chemical species. This is application reported in the website itself. So I would like to conclude here that pharmacoinformatics, whether it is based on chemoinformatics tools or molecular docking tools or artificial intelligence or simple quantum chemistry is not simple. It is very complicated, no doubt. But we are using the simplified version of quantum chemistry in drug discovery, like NBO analysis and the fluorine ion affinity, fluoride ion affinity, that kind of things. Using all that, we can get lots of information. The very first thing, the beginning of the entire thing, is to think in 3 dimensions. I'm sure those who do it will certainly succeed. Small as well as macromolecules, 3D objects. Rational design is possible. Incorporation of the fluorine-containing substituents has demonstrated remarkable influence on both pharmacokinetics and pharmacodynamic profiles. Strong electron withdrawing nature, high lipophilicity, and metabolic stability, and other related properties are being exploited in the medicinal chemistry. Most of these properties can be analyzed with the help of pharmacoinformatics. Of course, we have to give experimental validation towards the end of the project, but lots of homework can be done with the help of pharmacoinformatics before going into organic lab. These effects collectively contribute to greater chemical stability, reduced oxidative metabolism, and improved ligand receptor complementarity. That is what is the strength behind pharmacoinformatics in fluorine chemistry. These are some of the publications from our lab. This is the team which is working. I should agree that we are not focusing our work on fluorine chemistry, but we have several examples which contain fluorine as a substituent in our compounds. This entire presentation was compiled by all of them, mostly led by Ms. Rupal, and everybody contributed 2 or 3 slides in preparing this. So I thank all of them for this nice presentation. Thank you. Yes.
Thank you, sir, for very nice elaborative journey in the different parts. I was wondering, like the fluorine, like we are really enjoying that lecture. So you were saying that it also goes into the binding pockets. The hydrophobic, the fluorine interacts with the hydrophobic domain of the protein. So how does that happen? Because this fluorine, that is actually, it has strong electronegativity, right?
Right.
It has a little bit of polarity, kind of case. And in the hydrophobic pocket, when they go, what do you mean? Like you need to have a direct aromatic surface, which should be like a little bit.
Like leucine, isoleucine, and phenylalanine, that kind of amino acids, which form a sub-pocket inside the main pocket. And it is found that in our all our examples, that the CF3 containing units are majorly entering into that kind of sub-pocket inside the main pocket of the target. Like EGFR example, FTSZ example, and as well as in the artificial intelligence based docking when we did. Everywhere we have found that.
Also, like specific to aromatic side chain based amino acids, where like
Even if we have fluorophenyl group or difluoroamide group is present, this amide group is interacting in a hydrogen bonding fashion. But the two fluorines are not really forming any hydrogen bonding interaction. It's not exactly hydrophobic in the case of difluorophenyl difluoroamide group, but it is that fluorine we searched for that, and it's not forming any hydrogen bonding interaction with any of the NH centers. So that's what surprised for us also. But generally, we found that some of the discussion, even OCF3 group is also going into a hydrophobic pocket. So this was the observation.
In one of the papers which I was reading in my presentation, and which I showed, what I read, that fluorine, because of the very high electronegativity, it induces that CH bond neighboring to like in the pocket, there is CH bond, that CH bond gets polarized, and so CH becomes now the hydrogen donor.
okay.
So mostly that.
So that is like a London dispersion kind of things, the induced dipole.
Kind of. So that majority of the hydrogen bond by the fluorine compounds was with CH donor. And other than, but yeah, definitely there was NH and OH were also donors, but majority of the hydrogen bond with CH. So because that fluorine was inducing adjacent CH to fluorine negative. I would say that.
Several interactions on Friday morning. Like, I was curious about the details of that too, but I kind of fell from the structures. But I think that would be maybe we can relook at those crystal structures or docking models after.
Yes. Yeah, yes, certainly whenever we don't have all the crystal structures available, but certainly wherever it is available, it is very informative.
We are curious to understand specific receptor in the molecules in the physical database you want to work. Anything else?
Any other questions?
Maybe that lies the thing like another curious curiosity driven question is that I've seen that many of these fluorinated compounds they are pretty much biocompatible, and right, and then they have also shown that they have a very nice pharmacokinetic and pharmaco.
Yeah, that is a big advantage. In fact,
Big advantage.
as the molecule is floating in all body fluids and reaching the site of action, that is all pharmacokinetics. And if that is facilitated by our substitutions, it will be nice. A few examples we have seen today morning. Also, we have seen a few examples, and I am sure in the coming 3 days we are going to have many more examples of the same.
Right. I think with this I request for Salman to join so that we can celebrate Professor Sebastian. And Dr. Ashish, you also to join for a photo. Okay, so this ends today's talks. So tomorrow morning again we have a long day already planned. So tomorrow morning tomorrow is Wednesday. So and there is no class, right? So do you want to start with is it okay? 10 a.m. we should start or 9:30? Can we start? So Sarjan?
Whatever, yeah. Are they asking everybody?
Generally they have a 9 to 10 class and tomorrow they have off. So is it okay that we start at 9:30? Okay, and then maybe afternoon lecture also 12 to 1:30. So we can start with that. Maybe instead of 2:30 we can start at 2. So that will be done by 4. Right? So that is the small change and I will write it in the group. The time.